Hi class, and welcome to your next video on mitosis. So today we're going to talk about cell division in mitosis. And first off, we want to think about what each cell has and why it might need to divide. So cells divide for three reasons for growth. Everyone is constantly growing. Remember, that is a characteristic of life. For development, not only do children develop into adults, but adults develop stem cells into blood cells. And to repair, if you get a scrape on your knee or you break a bone, you're going to use the process of mitosis to repair your body. So, essential features of cell division are that we transmit a copy of the genetic information. It's actually a duplicate copy. There are no changes in it. And we transmit materials necessary for the cell to survive and use genetic information, meaning we're using those organelles and we're transmitting those organelles into a second cell. So, the really important part of this is the DNA. The DNA plays a huge role in any kind of cell. So the first phase that we look at is interphase. And during interphase, DNA molecules are extended in an uncondensed form, which is called chromatin. And the cell prepares to split. So it doubles the DNA and it doubles the organelles, because we need that same amount of DNA and the same DNA in the new cell. During the division phase of the cell cycle, or mitosis, DNA molecules condense into chromosomes prior to their division. So chromosomes are DNA that is tightly wound around proteins connected by a centromere. So we're going to look at that here in just a second. So this first picture here is the DNA in the double helix form, which many people know. This form right here is it condensing. And this condensing is around a protein called a histone. We then have the DNA that connects to this red dot, which is a centromere, and it condenses more and more and more. And then we start to get the shape of a duplicated chromosome. So right here at number 3, we have an unduplicated chromosome. Here we have a duplicated chromosome. So anytime we see it in the formation of an X, it is a duplicated chromosome. So you see the DNA double helix here. We wrap it around the histone, and then we get the individual chromatin, which is more here and here, but on the whole, it's a chromosome. So mitotic division is the division of a somatic cell or a body cell in eukaryotic organisms. So we're always going to be dividing these cells into our somatic cells, and a single cell divides into 2 identical daughter cells. There should be no difference between the two. So here you see kind of what's happening at the end of mitosis, and you see the one becoming 2 by pinching itself off. So there's 3 major processes in cell division: interphase, mitosis, and cytokinesis. So interphase is where the cell replicates and prepares to divide. So we're going to replicate our organelles, we're going to replicate the amount of cytoplasm, and we're going to replicate our DNA. In mitosis, we're going to divide the nucleus and the chromosomes into 2 separate cells, so that one cell is now becoming 2. And cytokinesis is where we totally split it. So we have 4 phases to mitosis. We have prophase, metaphase, anaphase, telophase, in that order. And then at the very end, we can go through cytokinesis. It is not necessary to go through cytokinesis for mitotic divisions. So in prophase, the chromatin condenses into chromosomes. So remember, in interphase, we had all that chromatin. Now we're condensing it into a chromosome. And there's these things called spindle fibers, which form and attach to the chromosomes. They come from the centrioles of the cell. And then our nuclear envelope, remember, that surrounds our nucleus, breaks down. So right here we see the very, very start of prophase. We see our spindle fibers coming off of our centrioles and our chromatin condensing into chromosomes. And the dotted line here around the nucleus is the fact that our nuclear envelope is dissolving. In metaphase, the next phase, the chromosomes align along the equator of the cell. So our nuclear envelope has totally dissolved. Our centrioles have made those spindle fibers and they have attached to the chromosomes, and now they're all aligned along the center of the cell. Our next phase, anaphase, the chromosomes are pulled apart into sister chromatids. So we started with chromatin, then we duplicated it and made it chromosomes, and it was tightly wound. And now we're going to chromatids. So the spindle fibers attached to the centromeres shorten and pull the chromatids towards opposite poles. So one's going to one side, the other part of it is going to the other side. And we free the spindle fibers and they lengthen and push poles of the cell apart. So if we look here, what used to be an X or a chromosome is now a sister chromatid, and it's going towards the side or the pole that it belongs to. So in anaphase, you're pulling that apart. The last phase here is telophase, and the spindle fibers disintegrate. The nuclear envelope forms around both groups of chromosomes. Chromosomes revert to their extended state, which is chromatin, so that unwound state, and cytokinesis occurs. Cytokinesis, however, does not necessarily have to occur. If cytokinesis does not occur, we have two nuclei now, but we do not have two separate cells. So when cytokinesis occurs, it encloses each daughter nucleus into a separate cell, which becomes our sister, our daughter cells. So if we look here, we see what's called a cleavage furrow here, and we are splitting it into our two daughter cells. So make sure you have great notes and great pictures from this presentation, and we'll go over it next time in class.